The measure of intelligence is the ability to change.
Hi all, Dr. Clark here, and this lecture we're going to talk about the Calvin cycle, or what sometimes is called the Calvin cycle. Sometimes it's called the dark cycle, the light independent reaction, and sometimes it's called, after these three individuals who came up with it, it's called the Calvin-Benson-Bassham cycle, otherwise known as the CBB cycle. Now, I don't want to dive too much into this, but this is a classic or fairly classic example of an issue with the Nobel Prize. Melvin Calvin, okay, and Andrew Benson and James Bassham, they worked together on developing the Calvin cycle, or developing how plants take in carbon dioxide and convert carbon dioxide into carbohydrates, into glucose. Okay? What is the process by which this occurs? These three individuals worked on that process. In fact, they published papers together. Numerous papers were published either as pairs or as a team, were published on the different processes of the Calvin cycle. And when it was first described, it was known as the Calvin-Benson-Bassham cycle. Well, something happened. I'm not going to pretend to know what the argument was about, what the fight was about, who did what, anything like that. But Benson and Bassham got forgot. They got left off. And Melvin Calvin won the Nobel Prize for the cycle. And so now we normally just call it the Calvin cycle after Melvin Calvin. Although I don't want individuals to forget Benson and Bassham, because their work was directly related to this cycle, and their work was, they were publishers, and they published many procedures and processes that have to do with the Calvin cycle. And in fact, all three individuals should have probably won the Nobel Prize, in my opinion. Okay? I just want to put that out there. That's where the word Calvin cycle comes from, is after this individual, Melvin Calvin, who the cycle is now named for. Although you will see it as the Calvin-Benson-Bassham cycle, or CBB cycle, every once in a while. All right, so let's dive into what is the Calvin cycle. The Calvin cycle is taking material from the light-dependent reaction, okay, the one we just covered, that has photosystem 2 and photosystem 1, and the products of those systems are ATP and NADPH. It's taking that material, utilizing the energy from that material to create chemical bond energy, or to create a molecule that can have the ability to be long-term stored. So it's a molecule that can have long-term storage. Okay? ATP is a great molecule. It's a great energy molecule. But I mentioned this before, adenosine triphosphate is a unstable molecule, in the sense that it wants to break apart. It wants to release its energy readily. And that's because you have 3 phosphate groups on this molecule stuck together. And each phosphate group is negatively charged. So it's like taking 3 negative batteries, or 3 negative magnets, sorry. You could take 3 negative batteries too, but it's not going to be really the same. But 3 negative magnets, and sticking the negative sides together. and you're holding them. You have to hold them together because they want to break apart. It's very similar to kind of ideology of ATP molecule. It's great for quick energy. Once it's created, it can be utilized, but it's not good for storage molecules. So the process of the light independent reaction, or the Calvin cycle, is to take that ATP and that NADPH and make it more stable, create a more stable energetic source. Okay? So ATP is needed to drive endergonic reactions. Endergonic reactions are reactions that need energy. Okay? They need energy in order to proceed. Remember, exorganic reactions are reactions that give off energy. So ATP drives endergonic reactions. NADPH is what we utilize as an electron carrier. It carries electrons from one place to another region. And those electrons can be utilized to build more bonds. The hydrogen that's on NADPH can be utilized to drive another bond, maybe a hydrogen bond or a covalent bond. It also could be used to drive a proton gradient. Okay? But this NADPH has what we call reducing power. Now, reducing, we'll get to this in a second. Reducing means, in chemical terms, that you're adding an electron to something. Okay? So reducing means that you're adding an electron to a molecule, okay, or to an atom. That is reduction. Okay? When you take the electron away, it's called oxidation. Okay? We'll look at this. We'll come back and check this out in a second. Okay? So the synthesis of new molecules is taking products from the light-dependent reaction and utilizing them in the light-independent reaction, or the Calvin-Benson-Bassham cycle, or the Calvin cycle. Okay, the reaction in doing this is often known as the C3 photosynthesis. Now, C4 photosynthesis and CAM photosynthesis also are other types of photosynthesis that utilize similar methods, but there are a little bit different. They're a little bit different in the light independent portion. Okay, we'll cover C3, C4, and CAM in a little bit. Okay, so the Calvin cycle occurs in three main stages. The first stage is carbon fixation. Taking carbon from carbon dioxide, okay, so the air is coming in through the stomata, and that carbon dioxide is being picked up, and the carbon from that carbon dioxide is being removed. So you have oxygen left, and again, oxygen is a waste product, so that oxygen can be expelled out of the leaf stomata, but that carbon is going to bind to a molecule, okay, and that molecule is called RUBP. Okay? RUBP is probably the most abundant protein on the planet, okay, and that carbon is going to bind to it. Then, through the expenditure of some energy, through the shuffling of bonds, eventually you make molecules that can be combined to form sugar, to form glucose. Okay, we're going to go through what that shuffling and phosphorylation and removing of bonds and breaking bonds looks like in a little bit. Okay. Then, because this is a cycle, after that sugar molecule is being made, or the predecessor to the sugar molecule, the monomer to the sugar molecule is being made, okay, you're going to reform your original molecule. So you're going to bind carbon to RUBP, you're going to shuffle everything around, make a sugar, and then you're going to reform RUBP so the cycle can keep going. Let's look at this. Okay? So the Calvin cycle is going to take in 3 carbon dioxide molecules. Those 3 carbon dioxide molecules are going to bind to 3 molecules of RUBP. That's going to form 6 molecules of 3-phosphoglycerate. Okay? There you can see that a phosphate is being added to these molecules. Okay? Then, like I said before, you have to expend energy. This energy is coming from the light-dependent reaction. So's the NADPH. These are coming from the light-dependent reactions. And you're going to take and you're going to shuffle this 6,3-phosphoglycerate into other molecules by expending energy, utilizing the electrons, removing the hydrogens, okay, and you're going to form different molecules. In this case, you're going to form 6 molecules of glyceraldehyde 3-phosphate. That glyceraldehyde 3-phosphate, one of those molecules is going to be utilized to form glucose. The other 5 are going to go back into the remainder of the cycle. ATP from the light-dependent reaction is going to be utilized again, so energy is going to be utilized again to take that 5-glyceraldehyde 3-phosphate and recombine it to form 3 molecules of RUBP. This is the Calvin-Benson-Bassham cycle. Okay? So this is that cycle that goes around, okay, and can pick up carbon dioxide again and keep that cycle going and going and going. Okay? Now, the interesting fact is it's not super efficient because the Calvin cycle has to turn 6 times in order to form a single glucose molecule. Because only 1 carbon is added from a carbon dioxide per turn. So this has to happen multiple times. And that multiple times that it's turning, you're burning ATP. You're burning NADPH to form that glucose molecule. Okay? Now, the nice thing is the energy that's utilized to form the ATP and the NADPH in the first place is free. It's coming from the sun. Okay? It's coming from photons. Okay? So, again, not very efficient because you got to burn a lot of energy to make that single glucose molecule. Now, we'll look at what that glucose molecule generates from an energy standpoint when we talk about cellular respiration. We'll look at how much energy you get from a glucose molecule. But you got to kick in a lot of energy to form that glucose molecule to begin with. The other cool thing about the Calvin cycle and photosynthesis in general is, remember when we were talking about the Calvin cycle a second ago, I said, yeah, it's burning ATP and NADPH. Well, the energy is being transferred. The electrons are being transferred. But the original substances, the original substrates, ADP and inorganic phosphate, is still around. And it goes back into the light-dependent reaction. NADP and hydrogen ions, in some cases, are still around, and they go back in to the light-dependent reaction. Okay, so the whole system is cyclic. Okay, so you have the Calvin-Benson-Bassham cycle, but it's also all cyclic in the sense that the reactants are being fed back into the light-dependent reaction. Okay, so again, just to recap that, ADP is available for chemiosmosis in photosystem 2. NADP goes back into the electron transport system for photosystem 1. All right, so let's look at this again just to kind of recap what's going on in the Calvin-Benson-Bassham cycle. Like I said before, Rubisco, or RUBP, is probably the most abundant protein on Earth. Now, we don't know all of them, but it's, it's fairly abundant. And I shouldn't say that. Where this is occurring, this fixation of RUBP to carbon dioxide, where that occurs, that fixation occurs at Rubisco, or the protein Rubisco allows for that fixation of RUBP in carbon to form 6,3-phosphoglycerate. Okay, so I don't want you to think RUBP is Rubisco. Okay, Rubisco is a protein. RUBP is a molecule that's going to be bound up with carbon dioxide, or with carbon from a carbon dioxide molecule, to form this. Okay, so that 6,3-phosphoglycerate, again, ATP is utilized to form a intermediate molecule, which we call 6,1,3 bis or bis phosphoglycerate. Okay? And then you're going to utilize the energy from that NADPH, okay, to form that 6 molecules of glyceraldehyde 3-phosphate. One's going to be donated to form glucose or other sugars like fructose. And then glucose molecules can be bound together to form starches, etc. That material is then regenerated. Again, ATP is utilized to reform RUBP. Okay? Now, sometimes you have to deal with cycles. You have to deal with material that's entering the cycle that doesn't belong. Okay? Or you have to deal with situations where you can't get the material that you need to go into the cycle. And that is what we call photorespiration. Okay, so we talked about this at the very beginning. Stomata, little pores on the bottom of a leaf, are opening up to allow for carbon dioxide to enter the tissue of the plant leaf and oxygen to leave the tissue of the plant. Well, anytime you open those stomata, you have the potential for water to be transpiring or go through the process of transpiration and leave the plant. Especially in hot regions or arid regions, this can be an extreme problem. You can't open your stomata, otherwise you'll lose too much water and you won't be able to do the light-dependent portion of the reaction because you can't split the water molecules to donate electrons. So you have to kind of, open the stomata, get as much carbon dioxide as fast as you can, risk losing a bunch of water, so you have to hold water in your tissue, or you have to generate another way to go about photosynthesis. And that's kind of what we're going to talk about. So, plants close and open their stomata or stoma. We already talked about this. Okay? If you close your stoma and prevent that gas exchange, oxygen builds up in the tissue. If oxygen builds up in the tissue, it binds to RUBP. Okay, so that Rubisco protein that grabs that or allows for that RUBP to come in and bind up to carbon dioxide, it also allows for oxygen to get stuck on the RUBP. if that oxygen gets stuck on the RUBP, okay, so if oxygen levels build up in the tissue of the leaves, then you get what we call photorespiration, okay, and it short circuits the Calvin cycle. It ends the Calvin cycle. Okay? And the organism cannot go through photosynthesis. Okay? So that Rubisco enzyme will fix oxygen instead of carbon. Okay? So again, like I said before, it's not a perfect process. Okay? You have ways that you can short circuit the process. So plants had to evolve and develop a way to prevent oxygen building up in their system and still allow for carbon dioxide to come in. So here's a diagram of that, okay, just to make it a little bit easier. Okay, again, this is the top portion of the leaf called the epidermis. Okay, and often there will be a cuticle layer that sits on top that prevents UV radiation damage and allows for water to not evaporate as fast, and damage to the leaf, damage from insects, lots of things. Okay, then you have an epidermal layer, and then you have some layers of leaves or layers of cells. But this is what I'm talking about here. These are called the parenchyma. This is called the spongy parenchyma. But this region has openings, and it allows for water to remove or come out of the system. At the same time, carbon dioxide would come in, oxygen would be leaving. That's when the stoma or the guard cells are open. If you do that, lots of water is going to come out. If you close it off so no water comes out, oxygen builds up in here. And you short circuit your system. You get photorespiration. No carbon dioxide can come in, oxygen builds up too much, and you start not being able to generate glucose, sugars, okay? And basically killing the system. Okay, so some plants evolved to perform photosynthesis by using what we call C4 photosynthesis. Okay, so some of these plants are different types of grasses. Okay, so sugarcane and even corn are also grasses, or they evolved from grass-like ancestors. So many grasses, sugarcanes, corns, other annual plants, often annual plants, okay, that occur in arid regions will use C4 photosynthesis. So what C4 photosynthesis is it's the light-dependent reaction is pretty much the same. It's the light-independent reaction. There's some extra steps that are added in. So instead of opening your stomata and allowing carbon dioxide to come in all the time, you can open your stomata, allow carbon dioxide to come in, and then fix that carbon dioxide into a different molecule, into a more stable molecule, and you can store that molecule for later use. So during hot weather, you don't have to open up and exchange carbon dioxide. So during cool temperatures where you're going to lose less water, you can open up, take in carbon dioxide, fix it as another molecule in a different set of tissue, and then when it's hot, arid, and you're going to lose a lot of water if you open your stomata, you just don't. And you utilize the molecules that you had already fixed, that you already stored. Okay? And so C4 plants have a different type of cell. Okay? And they're called the bundle sheath cells. And so we're going to look at this process. All right, so this is carbon fixation, or C4 plants. This is how they differ than C3 plants. Carbon dioxide comes in through the stomata, and it's picked up by phosphophenyl pyruvate. That phosphophenyl pyruvate, oxalate acetate, is formed. Okay, so the carbon from carbon dioxide is picked up and form an oxalate acetate molecule. That then can be converted into malate, and malate can be stored or utilized at a different time. Okay, so it can be stored in the mesophyll cells, and then the Calvin-Benson-Bassham cycle occurs in the bundle sheath cells. Okay, so that malate can then be used. The carbon from that malate can go right into the Calvin cycle. RUBP is going to pick it up. The whole system is going to be the same. When that malate loses a carbon, it produces pyruvate. Now, pyruvate is going to be very familiar to you because this is also the molecule that we use to convert glucose through a process called glycolysis. Pyruvate is the molecule that comes from that. And we'll come back to it. That pyruvate, though, is then regenerated. Now, again, you're going to have to expend some energy to regenerate it into phosphophenyl pyruvate. So, C4 plants, they have to utilize a little more ATP, but they lose water less readily. They go through that photophosphorylation, or they go through that photorespiration less often. They don't have as much issue with oxygen building up in the system because they can store carbon dioxide more readily. All right, now there's another way plants can do this, okay? And that is to start storing water. Okay? So you can store water and you can be known as a succulent. So cacti, pineapple, there's a lot of different plants that store water in their tissue. Most of those do a process which we call chrysalic acid metabolism. Okay, or these guys are called CAM plants. Okay. Now, CAM plants do photosynthesis in both processes. They use photosynthesis from C4 pathway at night. So they open their stomata, take in carbon dioxide, bind it, okay, store it as malate at night, and then during the day, when the sun's out, they can close up their stomata, they can generate ATP, NADPH from the light-dependent reaction, and they can start burning up some of that malate that was stored at night. And so, when you have C4 plants, they're doing it this way. CAM plants, they still do it all in the mesophyll cells. They don't have two separate types of cells. So they do both processes in the mesophyll cells. They do the C4 pathway, so converting carbon dioxide into malate, storing the malate, then using the carbon from the malate to run the Calvin cycle. Pyruvate is regenerated, okay? But they're doing it instead of in different tissues, they're doing it at different times of the day, same tissue. Okay? So with that, we're now going to talk about how carbohydrates, proteins, nucleic acids, fatty acids, how are they converted into energy? We talked about how the energy is made, or at least how carbohydrates are made. Now we're going to talk about, well, what if you're not an autotroph and you're a heterotroph, or how do even autotrophs take those carbohydrates and generate energy to drive work? Okay? That process is called cellular respiration, and that's what we're going to talk about next time.